Ready to quit?
Yes.
All right. Good evening, guys. Today we're gonna go over module 11, the use of genetics. Do anybody know what genetics are? Lauren, pull your mask down so we can hear it nice and loud.
DNA and traits.
Good job. DNA and traits. So in this module, you will learn about some of the amazing ways we use DNA technology, including in crime scenes investigations. So the standards for this lesson are Bio 3.31, interprets how DNA is used for comparison and identification of organisms. Bio 3.32 summarize how transgender organisms are engineered to benefit society. Bio 3.3 evaluate some of the ethical issues surrounding the use of DNA technology, including cloning, genetically modified organisms, stem cell research, and human genome project. So now we're gonna move to the pre-assessment. The pre-assessment, it doesn't count against you in your grade or anything. It just let us know what you know before we start in the section. So look at your pre-assessment, and the first question says, I'm gonna read the first question, then I'm gonna let you all do the other 9, which is 10. Do it on your own. Which of the following would most likely happen first when using a gel electrolysis? A, turn on the gel electrolysis. Use a light box of black light to see the bands of DNA. Restrict DNA with enzymes. What do y'all think the answer would be?
Restrict DNA with enzymes.
Okay. All right. Finish up the other 9 on your own and raise your hand when you're finished. Raise your hand like you got a word or something on her. Doing all right, Noah? Okay. Tamara, you okay? Lauren, you okay? How many more you got left, Noah? Tamara, how many more you got? All right, how many more you got, Lauren? Raise your hand when you're finished, and we'll move on to module 1, lesson 1, introduction. finished, Lauren? Okay. Look over it, make sure everything's like you want it. Finish, Lauren? Check over it. We'll move on as soon as Tamara finish. And she look over her paper. Alright, Tamara's finished. So now we're gonna move to Module 11, Lesson 1, Introduction. Lesson 1 is about genetic engineering. In Lesson 1, you will begin your study of genetic engineering. It's fascinating how scientists can manipulate change. DNA. Do anybody know what DNA stands for? So the next thing we're gonna do, the notes. No, we're gonna do the crossword puzzle first. So a blank is a picture of the chromosomes from the nucleus of an individual. 3 across. Anybody know what that is?
Karyotype?
Good job, Lauren. Karyotype. It's number 3 across. Karyotype.
Don't get that right.
No, do number 4 across. Read it. And it's what now? Recombinant DNA. Number 5, Tamara. Y'all read so everybody can hear you. Okay. Number 5, Human Genome Project was a research project completed by scientists to find the location of certain genes on the chromosome. Number 6, transgenic, an organism that contains the DNA of another species is called a what? That's transgenic. It's called a transgenic organism. TRANSGENIC, transgenic. Number 7 across. Anybody know, Lauren, what's number 7 across?
Gene therapy changes the cells so that they contain the correct DNA, which makes the correct protein needed to make the body work.
Good job. Genotherapy. So what's number 1 down? Anybody know what's number 1 down? Okay, Noah. Gel electrophorsis. GELECTROPHORESIC. Gel grow horses. Those are long words. Number 2 down. Restriction enzyme. A restriction enzyme cuts DNA in a specific place determined by the order of bases. RESTRICTIONENZYME, restriction enzyme. Okay, that's the crossword puzzle. Now, Module 11, Lesson 1 notes, genetic engineering. It say fill in the notes as you view the PowerPoint. Turn them in to your classroom teacher when complete. So go on your computer and go to your Module 11, Lesson 1 notes to your genetic engineer on your computer. Everybody there? Y'all on it? Okay. How are changes made to DNA? Go up there and read it and let me know what the answer is. Genetic engineering make changes in the genetic material, DNA of an organism to change the characteristics of the organism. Okay, everybody got that? Number 2, look in the picture. DNA from a what was inserted into a rabbit's DNA near the rabbit's fur blanket. So what did, in the picture, the DNA from a what? Jellyfish was inserted into a rabbit's DNA near the rabbit fur what? Gloves. Examples of genetic engineering. Taking a gene for poison in the tail of a blank and combining it with a blank gene. When insects eat the blank, the poison kills them. So in the first part, taking the gene from the poison in the tail of a what? Scorpion. And combining it with what? Cabbage. I wonder how they do that. Wonder how would they do that? What kind of tools y'all think they be using? Think they be using a needle or something like an insulin needle and stick it in the scorpion and then take out whatever they put in and put it in the cabbage. Y'all think that's how they do it? Possible. You right, Noah. Okay. Inserting human genes into what? Y'all gotta take them mess down so y'all won't be here, okay? So they produce milk that helps treat what? Lung diseases. A goat that has blank genes and creates silk in its milk. Has what kind of genes? Spider genes. I wonder how the word spider can live. How do a goat live with spider genes? What do y'all think a spider and a goat got in common? Huh? I wouldn't think so neither. A spider and a goat. A spider is an 8-legged animal and a goat is a 4-legged animal. A spider crawls and a goat walks around saying bad all day. And what does a spider say? It's kind of strange, don't you think so? Yeah. But hey, DNA, anything possible. Recombinant DNA. Recombinant DNA. Recombinant DNA involves taking what? What it involves? Taking what? Genes. Involves taking genes from one organism and combining it with a strain of genes from another. Okay, so look closer at the picture that they're showing you. You can see several different strands of DNA combined. Note how the base pairs match up. Y'all on the picture? So, how do we manipulate genes? Manipulate means to change or alter. DNA is too long to work with so restricted enzymes. So you're gonna cut the DNA into smaller pieces so it's easier to work with. We also need something called a what? Cloning vector, which is a carrier that transfers DNA from one organism to another. Plasmids are often used as cloning vectors. A plasmid is a ring of DNA found in what? Bacteria cells used as a cloning vector. Have y'all ever hear about anything being cloned? Can people clone people? Y'all haven't never seen on TV, just a TV show, but people clone people and make them look like other people. It was a movie. I can't think of the movie name. The name of the movie that they clone these people. Okay, results. Insulin can be made the way by transferring the human genes for insulin into a bacterium. The bacterial reproduction over and over and over again, you are made enough for the drug insulin. And what else do they use insulin for? What do people take insulin for? Diabetes. So do y'all think that insulin, what they using in this got anything to do with diabetes? You don't know. The bacteria reproduce over and over again, you are making enough for the drug insulin. So combining human insulin DNA plus bacterial DNA equals recombinant DNA. So you need to place this into a host called a what? Transgenic organism. Okay, 16, the blank is the transgenic organism. It will reproduce thousands of times. What will reduce thousands of times? Bacteria. How many minutes is it?
19.
So in summing up the little mini lesson we did on DNA. So DNA is what, Lauren? Okay, genes. Tell me, what makes changing in the genetic material, DNA, of an organism change the characteristics of the organism? Genetic engineering. Lauren, tell me something that you got out of the lesson that you didn't know. I can't hear you.
I understand about
The what now?
transgenic.
Transgenic part. So it's so much that goes on with DNA and everything. It's a lot of stuff that people don't know about DNA, but you know, it's like when children are born and people say the children are not theirs, you know, they have a DNA testing with the parent and the other person to see is that, see, do that child belong to that man or that child belongs to that, well, you know it belongs to the woman because the woman the one that had it. So to see, do the child belongs to the man that they say the father is, and stuff like that. And there's several ways you can test people for DNA. You can do it from by brushing their tongue, putting a string of hair, or blood sample. It's different ways that you can test for DNA to find out about people and different stuff. So that's our mini lesson for today, and we continue to finish up with this as the day go on. Thank y'all for participating. I appreciate it. And thank you for the fine job that you all did.